Hello, my name is Jurre Kamphorst. I'm the VP of MS Technology and Biomarkers at Olaris, and I'm excited to tell you more about the poster that we presented at ASMS, or the American Society for Mass Spectrometry, earlier this month in Houston, Texas. Here's that poster. The title is Highly Standardized Metabolomic Analysis of Clinical Samples Using Triple Quadrupole Mass Spectrometry. And what I'll do in this presentation is take you through each of these sections and figures in more detail. In a nutshell, what we have done, we developed a triple quadrupole mass spec-based metabolomics method. And so there are certain advantages of using this type of instrumentation over high-resolution instruments that are more commonly used in metabolomics. These include highly sensitive and accurate analyses, a large linear range, and also simplified data analysis. And so we've built a method allowing us to measure hundreds of metabolites in a single run in a very highly reproducible manner. And so this allows us to robustly measure clinical samples, think about time courses, and also samples from different batches that can then be easily compared to each other. So before I dive more into the details of this method, I want to take a little bit of a step back and tell you a little bit more about Olaris. So Olaris is on a mission to revolutionize how diseases are being diagnosed and treated. And for that, we built our CEREBRO platform. CEREBRO stands for Comprehensive Early Responsive Evaluation of Biomarkers Related to Outcomes, and it combines metabolomics with machine learning. On the metabolomic front, we use both NMR and also now mass spectrometry. And using this platform, we built predictive models to be able to detect disease and to evaluate treatment responses. So metabolomics has a very provides a very the ability to has the ability to provide very powerful insights throughout drug discovery and diagnostic discovery. Um, what I'm showing here is the various phases of drug development and associated biomarker analysis as well. And so what is interesting to note is that different types of methodologies are being used depending on the stage that you're in. And so in the earlier stage uh, of the drug discovery, methods tend to be more focused on broad metabolite coverage. And so here you're talking about high resolution instruments and NMR. This typically comes at the expense of reproducibility. Um, Toward later stage of the clinical trials, analyses tend to be much more quantitative and accurate, but this comes at the expense of being able to look at only a limited set of metabolites. And so what we have done at Olaris, using our combination of NMR and LC-MS, is that we are actually able to, in a very highly reproducible manner, measure hundreds of metabolites. And so essentially, we are able to provide accuracy and reproducibility at scale. So now I'll talk a little bit more about how we went about setting up this method. And so there's over 300,000 endogenous metabolites that have been reported that play various roles in human physiology and disease. And so what we've done is we went to the HMDP Human Metabolome database, looking for those metabolites that are endogenous and that have been detected and quantified in both urine, plasma, um, in either urine or plasma or both. Um, and so that's what is shown here on the right, having metabolites with various chemical classes. This gave us a number of around 1200, and based on that, we prioritized on the order of 370 metabolites. And it was based on a number of things. We wanted to have maximum representation across metabolic pathways. We wanted to have optimal coverage. The NMR covers about 50% of most classes, and so we wanted to improve coverage as well. But then we also wanted to have overlap with NMR for certain classes, specifically central metabolites, to serve as an orthogonal confirmation from our NMR findings as well. So then, based on a subset of those metabolites that we selected, the first step really was to identify the best LC column and also the best separation conditions. And so we wanted to focus more on the polar metabolites first, and that's why we decided to go with HILIC LCMS. And so there's various columns that we tested that are shown here, and they were tested with mobile phases, with pHs, with low, neutral, and high pH. We then introduced a scoring system, color coding essentially, looking at retention, peak quality, and signal intensity, giving those separately a green color if it was good, yellow if it's acceptable, and red if each of them was either of them was unacceptable. We then proceeded to essentially converge those separate metrics into one color per metabolite per condition. So we use the lowest score for each of those per metabolite per condition to gauge the overall performance. And so there you can see here there's three different columns that we used in the different mobile phase conditions. And what you can clearly see from here is that in our hands, the particular column that operated well was the BEH HILIC column in neutral conditions, where almost 60% of the metabolites that we looked for had good performance and 26 acceptable. And so what we decided to do is to move forward with this particular column, so the BEH HILIC column, for our further method development. We then finalized the gradient conditions, and then the next step was for us for all of the 370 some metabolites that we acquired, is to get various features. And so we very carefully built a SRM panel, and we're now able to annotate metabolites based on a combination of retention time that we acquired using the standards. We acquired 2 SRMs per metabolite, and so those separately, and also the ratio of the intensities from these SRMs are also features we can use for the annotation. And then finally, and I'll talk a little bit more about that in a second, for a subset of these metabolites, we also have concurrent or the corresponding frequency standards as well, that allows us for another mode of annotation. And so together with the ultra-fast SRM monitoring of the thermal altitude instrument that we're using, we're able to have 768 SRMs in a single 20-minute run, covering 373 unique metabolites. So, as I said before, we wanted to have a very accurate and reproducible platform. And so the use of 13C labeled isotopologues of indigenous metabolites are chemically very similar to the actual endogenous metabolites, but can be differentiated for MS. And so they are the perfect internal standards, which is the gold standard to use if you want to have accurate and quantitative information. And so the use of the internal standards reduces the technical variation due to sample handling and analysis, and also corrects for matrix effects. And so while getting individual standards for all of these metabolites is cost prohibitive, there's a workaround in the form of 13C yeast. And so these yeast cells are simply being grown on 13C glucose instead of the normal 12C glucose. And then the resulting 13C labeled yeast extract can be used to spike into your sample to have many 13C standards, and this is commercially available. And so we are using this, and we currently have 116 13C metabolites in the panel, and we'll identify more through a collaboration with Thermo using high-resolution mass spec, and also we are looking into using 13C isoforms of closely eluting metabolites as well as surrogate standards. And I'll talk a little bit more about that in a minute. So we performed very thorough validation of our methods. I want to highlight one particular experiment we've done here. So for our work, we are interested specifically in urine. Urine samples are very challenging due to the high salinity and also variation in specific gravity. And so what we've done here is we did an experiment with pooled urine that we either diluted or concentrated to cover it in the entirety of the specific gravity range, going from very dilute on the left end to very concentrated all the way on the right end of the spectrum. Um, and so what we did there is we spiked into each of the samples at the various concentrations. We spiked in the fertility yeast and prepped the samples and analyzed them by our methods. And so here you see four metabolites that are representative of various chemical classes. And what you can see going from very dilute all the way to very concentrated on the x-axis is that if you just look at the MS intensity without any sort of normalization, is that as the sample gets more concentrated, the linear signal actually, the signal drops off. And so what's happening here is that there should be a linear increase of the signal because you go from very concentrated sample to a very dilute sample to a very concentrated sample, was not what we were observing, and that's due to the high presence of the salts causing ion suppression. However, if we use their respective standards and essentially express the metabolite intensity as a ratio to their fertility standards, you can see that now we have a very linear signal with R squared in the 0.99 and higher, showing you that across a wide range, in this case over 30-fold range of specific gravity, we maintain a linear signal. Diving a little bit more into detail about this, one of the potential concerns with fertility yeast is that the levels of metabolites present in yeast can be very different than what you'll observe in human urine samples. And so to see if that was a concern, we looked for one of the extreme examples, in this case for carnitine. And so on the left, you see an example of the peak for the endogenous molecule, and on the right, for its fertility standard coming from the yeast. And so we wanted to see, even with those huge intensity differences, with the signal in urine being on the order of millions, and for the fertility standard only in the thousands, if we can still maintain a linear signal using this big difference in intensities. And so what you can see is that even with extreme faults, extreme ratios in the thousands, we're able to maintain a linear range as well, which I think speaks to the quality of the analysis using our instrument. The other thing that I briefly mentioned already before is that we don't have fertility standards for all of our metabolites, but in those cases, we are looking to use the standards for closely matching and closely eluting metabolites as a surrogate standard. And this is an example of N-acetylvaline, which has a retention time of 5.8. And then in this case, we used fertility labeled tryptophan, which has a retention time of almost 5.7, so pretty closely eluting as its internal standard. And what you can see here is that that also leads to a quite linear response over the entire range of the specific gravities that we tested. So kind of bringing this all back, what I'm showing here is a network of of molecules based on chemical similarity, and then color-coded based on if they are measured by NMR, our LC-MS panel in teal, or both in red. And what you can see here is the current network of those 1200 molecules I was talking about before, and what the coverage is. And so it's clear that we have various areas that we are covering pretty well, and this panel is going to evolve. And then there's also areas where there's still almost no coverage. Those areas are actually the more lipophilic substances, the lipids, for example, and so this is an area for us to work on in the future. So this slide then summarizes our method. So we're using a triple quadrupole based metabolomics method, and so our polar metabolite workflow requires minimal simple volumes. 25 milliliters for urine, a 20-minute run time, and it uses semi-automated processing using Skyline. We also have an in-house generated QC and test pipeline that we use to further process and analyze the data. Future development will include methods for non-polar compounds, including lipids. And so collectively, the data that's shown here demonstrates that the Olaris triple quad panel, together also with our NMR method as well, enables accuracy and reproducibility at scale. Thank you for your time, and if you're interested to learn more about Olaris, please check us out on the website. Thank you.